reached out by various people to give this talk. I think it's because some people at least think I'm running a good lab, and a lot of people expressed interest in this. So I will not speak a lot about science today. Most of this requires significant background, and you know, the more I go into the detail, which and details are actually important, the more you will actually start, you know, switching off. So instead, I just want to briefly introduce myself, my team, what we are doing, which way we are going, what are the opportunities, and so forth. So the lab, you know, before, and maybe some people will disagree with me, but before we had a, oh, Musa Edigenov says hello. Okay, hi Musa. So before, I think it was late 2018 when we could actually started to move into C4 building. Before that, scientific projects at NU, there were some, okay? Some people were lucky to get some lab space, but vast majority of professors had zero chance of actually accomplishing anything. Right now, everything is fine. You're looking at my lab. I'm sharing it with Anna Andreeva from biology department. And you see, actually, you can see her in a background doing something. This photo is fresh, taken a couple days ago. And we share the lab. Of course, there are some fights about space and everything, but we can handle this because we are also related to each other. So that helps. Now, the room is small. Okay, that's a picture taken from, you know, from the opposite wall, essentially. And Anna is almost standing at opposite you know, across the room. So it's not a large space, and that's a picture taken from a different point in the same room. The room itself is small. We cannot physically accommodate a lot of people, and she has a few, I don't know, 3, 4 people working with her, and I have maybe 5 or 6 because I have a larger project compared to her. I have simply more money, and also accordingly more responsibilities and more goals in the project, which I sign up to. Essentially, I said, okay, I will do this and that. And after the project is over, I have to deliver the results. So, so you see the lab is, I don't know if you can tell from this or not, but the lab itself, it has a modern furniture. It's a lab, it's equipped really, really well. It's lab furniture from actually German company which specializes in this, and it's the same throughout the whole C4 building. It's top-notch. And because we are quite well funded, we have all the little things which are required for everyday work. It's been you know, quite painful to acquire everything from glassware to plastic to pipettes, all kinds of different things. I think right now we are okay. There is some equipment which is located in the lab, like on the bottom right corner, you see a little bit of a actually very large ultracentrifuge. It's extremely expensive equipment, which I got more or less for free. You know, long story, but and there are other things as well, like for example, nanodrop, which is extremely handy in any lab. It's quite amazing that need a couple microliters to actually record a full spectrum of pretty much anything. So the technology, it's being developed with amazing speed. On the back, you see like a biosafety cabinet to work with cells, and that's mostly my wife who is doing this kind of work. And on some project, we actually do collaborate. We, she helps me with cell biology if I need anything. And if she needs proteins, I make them for her. Okay, and that's been, it worked actually quite well in the past. So we probably will be we'll keep doing this in the next couple of years. I'm pretty sure about that. So now we also have a very a good equipment scattered across the neighboring labs, which we can access, at least in theory, because the provision upon which we got everything is that we've been told, okay, you guys need to share everything with other people. Of course, some of the equipment is kind of very specific, and people consider that equipment their own, and it's may not be easy. It depends a lot on personality of a PI, okay, whether a PI is willing to share the equipment or not, but usually enough pressure can be made to actually persuade almost anyone to share, because really it's all the university money. We get almost all of the equipment is purchased with Nazarbayev University money. And this equipment you see here is actually really shared because it's in a common room, doesn't belong to anybody. This is actually a plate reader which people brought from block 7. Block 7 is now more or less empty. Nothing's going on in block 7, and it's a reasonable plate reader. I don't know how long it will last because it's been used so heavily in the last, I don't know, 6-7 years that I don't know. But so far so good. We have also gel scanner, which can scan any gel, literally any gel, and gives fantastic results, publication quality. And on the right, you see FPLC, which is a protein chromatography system. You can purify proteins with that, and it's almost just me who is actually using it. So you cannot do science without money. And I've been funded reasonably well over the past 5 or 6 years. Almost immediately when I got here, I got a grant, which was at that time a standard grant for, it's called ORAO. And it's standard, it's $100,000 per year for 3 years. This was completed last year. Actually, it was completed already, let's say 1 year ago. And now I have another grant, which is the biggest available at NU, I think. Maybe there are bigger ones, but this is actually quite big. At that time, I think there was about 21 of those awarded for new professors, and the chances of getting it wasn't actually bad. I got it. Many other people got it as well. But that was probably like the last big chunk of money which flew into the funding system, because right now it's more difficult. But still, just like a little remark, it's actually quite, in my experience, again, my experience was that it's much easier to get funding at NU compared to Western universities. So I have a lot of money. Some of it goes to people. For example, postdocs are paid really well. I have one postdoc, Darkhan Utepbergenov. I have Ayjan Kirova, who is a PhD student. I have one master student who is also our alumni. So he just finished, completed bachelor's degree of chemistry department last year. I have 3 students at the moment who are undergraduate students, 4th year and 1 4th year and to third-year students. And of course, there are collaborators in the same room. Of course, there are students from Anna Andreeva. I also collaborate with National Biotechnology Center. Sometimes they come to the lab to do some little things. But of course, everything changed with COVID. Now, as you know, lab access is very restricted. I think in the past couple of weeks, the regulations are that there would be between 1 and 2 people allowed at the same time in a single room. So it depends on the square footage of the lab, but this is a nightmare. Actually, you can't do any project very efficiently if you have, let's say, 1 person working on it. And but that's the reality we are living in, and let's hope that things will get better. But I did have, let's say, if you look at third-year students, Janibek and Azad, I sent them home. Actually, they sent themselves home because it's just there is no way for them to work continuously on this. They just cannot legally access the lab because of the restrictions. All right, so of course, I'll talk about research. I study in general several topics which are kind of interconnected with each other. There is oxidative stress, glycative stress, which if you don't know what it is, I'll explain it in a second. I am generally interested in enzymology and protein chemistry. So if you think of a person who kind of works with proteins, can make them, can study them, and knows a lot about them, that would be me. Okay, maybe there is 1 or 2 people in biology department who will have some expertise as well, but you know, and definitely they know a lot as well. But I have probably a little bit more knowledge on chemistry side of this. So if you have some ideas, some questions about proteins, I would be the one to ask, I think, at NU at least. And so I also kind of recently got interested in using enzymes in organic synthesis. This, of course, is quite an old idea, but still there are many examples, and the whole thing now develops really quickly. And I got interested and tried to do one thing or another, but this is like a side project more or less, but I will also mention it today. So what I'm doing, and I wanted to talk about this glycative stress. I think it's quite interesting and important. And this is kind of an introductory slide. If you go to a clinical lab, you can get a test for glycated hemoglobin. So glycated hemoglobin is a molecule. You probably know what hemoglobin is. The molecule that reacted with glucose. It's a spontaneous chemical reaction. There is no way to avoid it. Every person has some glycated protein. Okay, so it's a direct reaction of glucose with the protein called hemoglobin. You know what it does. Quite important for all kinds of, it actually, of course, transports oxygen, but the ramifications of, let's say, excess of glycation can be, for example, damaging kidney, and which, probably not very easy to figure out right away from its function, but that's what people have been actually working on for quite a long time. And it's now quite clear that, let's say you get diabetes. In diabetes, the hallmark of diabetes would be glycation of hemoglobin. So let's say you have a severe diabetes, 6.5% of your hemoglobin will be reacted with glucose. and it doesn't function properly anymore. And when the cell starts degrading it, it doesn't also degrade properly, and all kinds of pathological conditions arise. So that's why you probably, all of you have heard that sugars are bad, and that's exactly why they're bad. It's not that you have a lot of calories and you get fat. Yes, that's one thing, but the real problem, and the biggest problem, is actually chemical reactivity of sugar itself. It will react no matter what. It just has a built-in property, or let's say capacity, to react with, pretty much any nucleophile. And since there is so much hemoglobin in blood, that would be the first thing to react with. And so even worse is another molecule, which is a product of glucose decomposition. Let's say you decompose glucose in glycolases. If everything goes well, you get this tricarbon intermediate. But there is a side product, which approximately 1% of glucose, up to 1%, let's say, goes to methylglyoxal. This molecule, which you see on the top, just a moment, methylglyoxal, okay, this guy. This is a product of glucose metabolism. Every single organism has it. It's unavoidable. As you see, there are two carbonyl groups. They're highly reactive. And one of the products, for example, when it reacts with arginine residues, is this hydroimidazolone derivative, which, has nothing to do with, natural amino acid. And if you have a lot of those, the protein will probably stop functioning properly, and there are all kinds of bad things which going to happen. And again, usually this is kidney, but there are also some consequences for brain and some other things. All right. So, and that's why all of the organisms, living organisms, have a very efficient system for detoxification of methylglyoxal. And as you see, it's kind of a disproportionation reaction. You have two carbonyl groups, and it's kind of like an intermolecular reaction. Those of you who have heard of organic chemistry, it's like an intermolecular Cannizzaro reaction, where essentially one carbon atom gets oxidized, another gets reduced, and in the end, you get simply lactic acid, shown here. All right, so that's the same reaction one more time. It's actually, so this is methylglyoxal. There are several enzymes which can do this, catalyze this reaction. Those are called glyoxylases. And they are quite efficient. And this lactic acid is, of course, harmless, absolutely unreactive. It's a normal product of metabolism. You will not be able to harm yourself with this one. But this guy, methylglyoxal, is really bad. Now, what I study is glyoxylases in general, and there have been several of them discovered relatively recently. They are prominently present in yeast and bacteria and in plants. In humans, there is like this classical system of detoxification, which involves glyoxylase 1 and 2, here, shown here, but they require glutathione. And, this is kind of, people knew this about them for more than 60 years, okay? This is very well established, but recently there is another enzyme which discovered in 95. Recently people have actually characterized them, and actually they have found that it doesn't require glutathione, and it inactivates methylglyoxal in one step. As I said, they are present in plants and bacteria and yeast, or generally in fungi, but in humans there is only one so far, only single protein, which is called DG1, okay, shown here, well, written here, that's the name. The thing about DG1 is actually it does catalyze this reaction, but it's actually very, very inefficient. And the question is why you would have something like that in your body, which is not very effective, okay? So it catalyzes, let's say, less than one catalytic act per second, okay? Which is, so in other words, its kcat is 1 second, 1 reverse second, which is extremely low. So there is a big question, why the hell do we need this in our body? So probably it does something else, we don't know. But the thing is, if you don't have DJ1 in your genome, let's say it's got inactivated by mutation or something like that, there will be Parkinson's disease, all right, in these people. And right now there is enough evidence for that to be true. There may be, I don't know, about 8 to 10 different mutations have been isolated, all of them in DJ1, which lead to Parkinson's disease. It's not too bad because there are some genes which, when they are mutated, Parkinson occurs, let's say, at age of 20 on average. DJ1, if it's mutated, and of course these are all germline mutations. Okay, so let's say you have and it's a recessive mutation, so you need to have both copies of DJ1 inactivated, and if that happens, then DJ1, these people get Parkinson at about 46 years old. Okay, and that's no fun because there is no cure for Parkinson. People learn how to slow it down. You can slow it down, but it's not curable disease. It's a question when. And another thing about Parkinson is that this deterioration, basically the symptoms onset and everything, it's slow, but it's constant. Even if you take the drugs like levodopa and other things, it's just constant decline. And you can almost, be sure that, yes, you have 5 years, maybe 7 years, the quality of life starts to decrease immediately. And it's just, now the question is how much can you take? And so now there were some indications. So people have actually said, okay, if DJ1 cannot do this reaction efficiently, maybe it does something else. And some people came up with the idea that it actually catalyzes slightly different reactions. In other words, the methylglyoxal shown here reacts with nucleophiles like thiol and amino groups on the protein and forms these it's called hemisiwas, stalhemiaminals, these kind of products. And of course, those are not, this is also not fun. You modify your protein heavily with this, extra entities, and they definitely would lead to some decline in function. So now there have been indications that DJ1 actually can come to rescue and catalyze and work on these modified proteins and DNA, those things being substrate, and DJ1 can actually release this lactate back. So essentially it doesn't work on methylglyoxal directly, but actually works on these proteins, modified proteins. So there were 2, or actually more, maybe like 5 or 6 papers like that. And that's where we started our work. So this is, we published this already, what is it, a year ago, a little bit more than a year ago, December 2019. So that's a professor from Nazarbayev University. That's another one, okay? And the rest are undergrads. So they have been helping a lot. Mostly those are bio people, but Zhenibek is actually was, probably most helpful. He probably did like at least a half, but because Dr. Andreeva used, extremely sophisticated things like CRISPR and things like that, and it took her like half a year to establish cell lines and everything. She deservedly is the first author on this. But almost everything else, Janibek kind of spearheaded this effort and was extremely efficient as a chemistry student in developing mathematical models and all kind of different things. So this is Journal of Biological Chemistry, guys. You should actually, one of the things you should learn very, you should understand the quality of a journal very well. So impact factor definitely helps, but you should how to say, look at other things as well. So, and my favorite metrics for journals quality is not impact factor, because it's easily manipulated by all kinds of different people. It's scientific journal rank, so SGR, okay, and anything close to 1 and above is actually good. Very, let's say it's if 0.5 and below, this is actually already questionable, okay? So JBC, for example, it's 2.4. And of course, there are better journals. I don't know if you take Nature is probably something like 11, or if you take Angewandte Chemie, which is best journal for chemists, is probably something like 7 or 8. So everything is relative, but I think on this one we did a pretty good job. I will not go into the details, but I just want to show you a couple of things. We do use a lot of different methods, and it's quite, I'm actually quite happy that so many different methods are available. So this is, for example, mass spectrometry on the intact protein, which was done at National Biotechnology Center. They have a high-end mass spec for proteins. This is chromatography for protein chromatography, right? I showed you the equipment. So now we can do this. And this is all for protein characterization. This is melting point of a protein determined actually by a real-time PCR machine. It's usually used for real-time PCR, but you can also measure melting point of a protein with it. This is NMR, okay, on the left. So we have NMR in chemistry department. Now it belongs to Office of Provost, but chemistry department is uses 99% of NMR time. So what's this is some optical test, and this is what Anna Andreeva has been doing. So if she actually established a CRISPR, so she used CRISPR technology. Of course, I hope most of you have heard about this. It's genome editing, and she got rid of DG1 in cells. Okay, it's a very nice. You see, it's almost here as if you took a razor and removed DG1 from cells, right? So it almost looks like a magic to me. And but now technology, all kinds of technologies allow to do that. We also confirmed it with sequencing of genomic DNA. Everything went really well at that time. So it was extremely important contribution to that paper. Anyway, we were able to figure out that DG1 is not a deglycase. It's something else. It must be doing something else. We characterized hemethioacetals equilibrium. Of course, I showed you like maybe 20% of that paper. As I said, I don't want to go into the details. I just wanted to highlight some of the methods we are using. And yeah, that's conclusion. So there are other things we are doing at the moment. Of course, this is the past. This is we are done with it. Okay, so I sent off a paper last week. Hopefully it get accepted. And a couple other things. For example, you can use glyoxylases in synthetic chemistry. We still, oh, here, this is not finished. Okay, I have to, I'll tell you what it is. So molecular function of DG1 is still not known. We don't know what it does. Okay, we're trying to figure it out, but of course, it is very hard. People throughout the world are essentially trying to do the same things. Many of them are working for more than 10 years on this. Of course, you know, it's not like I come here, work 1 or 2 years and figure it out myself. Okay, this is its function. It's extremely hard. But of course, this is a kind of scientific project I like working on because, you know, simpler things are usually boring. And new protein tags, and I didn't finish, I guess I fell asleep yesterday or something. It's new protein tags for purification, okay? So we are experimenting. So the classical protein tag is, for example, a polyhistidine tags. We're now trying to use polyarginine. Of course, it's an old idea, but it worked surprisingly well on some of the proteins, and we kind of now trying to push it a little bit. Maybe, just maybe, it will work very well so that actually people will switch to it in the future. So yeah, this is, we are done with this, and synthetic chemistry. So this is like, let's say this is a method to measure glyoxylase activity. Okay, I told you about what glyoxylases do, right? And how do we measure activity? Well, we found a method to do that, and we described it. And this is DNPH assay, is classical assay, but it's discontinuous. Essentially, you need to take point at certain time, and then make a chemical reaction. Here, it's continuous, the red line. It's actually UV absorbance over time, makes it gazillion times easier. I hope people will be using it. Now, the synthetic chemistry application is actually extremely simple. You can take as R almost anything, and surprisingly, those glyoxylases I have in the lab, they actually will catalyze this reaction. Moreover, the reaction itself is boring. Actually, if you put this thing into alkaline solution, it will actually give you alpha hydroxycarboxylic acids as shown here, but it will be a racemic mixture. What we want is to get essentially stereo, how to say, we want to get enantiomer separately, right? So if, let's say, I take this variation of DG1 protein, which comes from plant, I can get, for example, L form of this alpha hydroxycarboxy acid. And if I take another one, I may be able to get a D form. Okay, it works in principle. There are difficulties with this, and the major disappointment as of recently was that actually if we take aromatic compounds, and that compounds which we were mostly interested in, they also give us pretty much racemic mixtures, unfortunately. And that's probably has to do with conjugation here. You see that this thing is conjugated and flat, and this rotation is probably limited, and that's why I guess we are getting racemic mixtures. With aliphatic compounds, it works actually quite well. And we will see. Maybe it will still work. We are kind of in the process of figuring that out. And in CRP grant, I have a couple of tasks. Probably most interesting has been, like, ever since I came here, is this when I purify, and it's actually maybe the last slide here. When I purify DG1 from bacterial cells, I can purify it. This DG1, you know, remember this is a protein which causes Parkinson, or actually its absence causes Parkinson. When I purify it on nickel column, when I put a his tag on it, like every protein in the world would behave like the protein, like its mutant, where I inactivate a residue in the active center. That's how it behaves. The column initially turns white. So this is nickel, it's blue, or let's say cyan color. And because there is so much protein on the column, it turns white. It kind of blocks the nickel sites. And then when you elute it, yeah, it goes back to cyan again. And eluted fractions are, sorry, this is wrong. Okay, this is wrong, disregarded. So this is, of course, correct. So you elute it right away with the imidazole by competition. It's easy to do. That's how much protein you get. It's a lot of protein. It's pure. Everything is nice. But if you work with wild type DG1, what happens is that the column turns into this peculiar blue-violet color, and it's a protein itself. Okay, we were able to show that the protein, or at least a complex with nickel, is responsible for this color. Moreover, the protein now has very strong affinity to the column. So you let's say here, all the protein is already gone, but as we increase the imidazole concentration, it still kind of clings to column. It's really hard to remove. And only at extremely high molar, 2 molar, 3 molar concentration of imidazole, we are able to get all of the protein eluted. So we don't know what it is. It's extremely interesting and at the same time difficult to figure it out. Maybe you guys may have some ideas how to do that. The thing is, when you elute this protein, let's say you are able to elute it with 3 molar imidazole, you can put it back on a column. Okay, let's say you can dialyze imidazole away and put it back, and now it behaves normal. Okay, so there is something which is kind of, how to say, transient. Okay, there is a transient modification, probably some high energy state of the protein, which makes it sort of like violet. But once you elute it and reapply onto the columns, this property is lost. And it's been a challenge for the last, I don't know, 3-4 years. We've been working on this. But still, we are trying to solve this because I'm pretty convinced that this property actually does have something to do with its real function. Okay, why the hell would the protein do something like that? It's the only protein in the world. There are millions of proteins, and DG1 is the only protein which does something like that. Okay, so but figuring essentially the molecular background of what it is extremely difficult. But it's let's say it's a high risk, high reward price. Well, I got money for this, so I'm gonna do it anyway. Let's say I'll try to figure it out. So anyway, I didn't want to, as I said, I didn't want to spend too much time on scientific part. I can answer any of the questions you have. Some people know me quite well, others don't. So you guys go ahead and ask questions. I think that's why you are here, and that's why I'm here.
Okay, so any of participants who wants to ask questions, they can turn on their microphones or write questions to the chat. Don't be shy. This is a good opportunity.
No? Okay, so I think one of the questions people ask me, what kind of students I try to recruit and how they end up in my lab. So it's been my experience that it's best, everything works best if I select students myself. And I don't like try to specifically kind of observe every single student in my class when I, for example, teach Gen Chem. It kind of comes natural when some people are more visible than others. And of course, I pay attention to the academic performance. Gen Chem, many of you probably will disagree or get some extremely negative emotions when I say that, but Gen Chem is generally a test for IQ and discipline at the same time. So you need both. So maybe you have IQ, high IQ, but if you lack discipline, I don't know. It's gonna you'll not get an A, not in my class anyway. And so I look at people. Sometimes it's not necessarily that I just there is a class and I take. I am determined to take a person from that class. Sometimes I select nobody, and sometimes I just want to take like half of the class. It just depends. Okay, so, but when people approach me and ask me if they can join the lab, well, that usually works not so well. I don't know the person. I don't know, or I know that probably this person would be not a good match. So I often say yes, but maybe like 30-40% of time I say yes, but I usually, it's not like I regret this, but it's not very efficient, let's say, okay, when that happens. So somebody asked, thank you, it was okay, Olga, you're welcome. Well, at least 20, there are 21, 5% of people found this presentation lively and interesting. So, okay.
Okay, maybe I can ask you some general questions about research that people also might be interested in. So, for example, how do you react when students make mistakes during the working process? So let's say you have some.
Oh, I am. Despite you know, in the class is a one thing, but in the lab I am extremely soft. Okay, so I never like. So you can ask anyone. So I'm kind of say, okay, fine. You just if you want, you repeat it. If you not, it's fine. I'll repeat it myself or something like that. You know.
Okay.
No, it's really it's not a
People
problem
ask,
at all.
how many hours do you spend in the lab per day?
Yeah, one thing is actually it's quite interesting. You go to sixth floor. Okay, most of you are bio people. You go to sixth floor. People you find in labs, there are 3 or 4 from all of you professors. It's me, Anna, Dos, and Alena. Okay, everyone else, yeah, and you put it on YouTube. Probably I don't care. Yeah, so I spent, I don't know, per day, 2 or 3 hours. I rarely see. Of course, there are some people coming and going, but for so far, and of course, depends on the year and COVID, everything. But so far, 6th floor is empty, and it's been empty even pre-COVID. Okay, there are not many people who are active on professor's side, and let me correct myself. Trifam, he is always there. He takes care of his flies. He doesn't have a choice. Other people, and of course, some people, for example, have extra rooms, let's say on 5th floor. So I don't, let's say, and on the other hand, if, for example, this can also reflect on my, let's say, my shortcomings. Maybe some people are able to organize everything in the lab so perfectly that they don't have to show up there. Okay, that's also possible. So, but I spent 2-3 hours. Sometimes I work the whole day. But usually that must be a weekend. But on average 2 or 3 hours. But it's true that the professor doesn't have to be there. Okay, especially if he or she has postdocs. But it's often that it's hard to find a good postdoc here, almost impossible. And that's why the professor should be in the lab at least, a couple times a week. Okay, that's my personal view of this.
Okay. Okay, I see another question. Is there a problem with a lack of reagents, tools, etc.? And if there is, how do you solve it?
Not for me. Of course, so I am, the last thing I did before joining NU is protein crystallography, and there is no way I can get a synchrotron time in Kazakhstan. Okay, the synchrotron, the nearest one of a good quality is in Europe. Even those ones which are in Moscow, or maybe in China, I don't know, it doesn't matter. I will never get there. So I had to switch to something else, and I did. So I'm now doing more, let's say, sort of like a biochemical projects. And yeah, so I kind of designed my project in such a way that there will be no lack of things. Okay, I think ahead and kind of design a project. But some other people do have lots of constraints. And if you take, for example, Anna Andreeva, she is, what would you call her? Geneticist, probably. And she needs mice. And that has been extremely difficult to arrange. All right, she's, in the end, she gets some mice from National Biotechnology Center, but it's extremely difficult on every level, from paperwork to actually getting mice. She lacks a good quality dissection microscope, all kinds of different things. My science is extremely simple, but her science is it's almost an opposite. She isolates embryos from mice, and then on embryo, which itself is like, I don't know, 4 millimeters, right? Something like that. She cuts out not ear, but inner ear, okay, with her hands under the microscope. So it's quite, so she has a quite elaborate model system. And of course, it requires a lot of different things. And yeah, she has difficulties. Right now, both of our microscopes, high-end like confocal microscopes, are broken. So that doesn't help us either. Okay, so in her case, mass spec is broken. It was up and running. There is one in National Biotechnology Center for proteins, but there is like a generic one at research, how they call, shared facility, and it's been up and running for like, I don't know, 2 months. It got fixed, but now it's broken again. So there are difficulties as well, of course, many difficulties. Yeah, but because of funding, because I, got funded, it was serious money. Some problems can be solved with money, yes, but some cannot be. So depends.
Do our professors have opportunity to travel between laboratories? So as you said, we don't have special equipment in Kazakhstan or anywhere near. Can you like travel to some country or to some other laboratory?
Some people do exclusively that. Okay, they say, okay, it's so difficult to work here with starting from organizing everything to actually getting space or getting some qualified help, that they actually go abroad and do everything abroad. But that's probably, and I completely understand that, but that's probably long term is not a good solution, because what's the point? I mean, if we are serious in kind of organizing some real research here, right? Going abroad and do research is not helping much. What I would try doing, and I sometimes do that, I would send a student abroad. Okay, and again, money on grant allows doing that. And of course, everyone is more than willing to go to US or Germany, but that is a different story. Okay, the student learns something and comes back. Okay, but yeah, so I guess I answered your question. I myself never went abroad, but some people do that a lot.
Okay, thank you very much. Another question also about students. So what do you think is the most popular misconceptions that students get regarding the research activity?
Oh, you guys have a very good questions. So there are many misconceptions, and well, it's not a misconception. It's probably like an attitude. Um, Often a student comes to a lab and automatically assumes that that he or she will be helpful and that he or she can contribute immediately something. And they get like, let's say 2 weeks passes and not much is happening in the lab and they get kind of a little bit frustrated. But the thing is, if it's a real research, first of all, 90% of experiment goes directly to trash. Otherwise, we would have already discovered everything. Okay, of course, there are projects where you go and measure some, I don't know, like air pollution or water pollution or something like that. And no matter what you do, you will get some data and you will publish it. But it's not the kind of research I'm doing. So very often students the misconception is essentially that they immediately can contribute something. Whereas in my case, and for example, in Anna's case as well, we need to invest a lot of time into students first for them to understand what they're doing and actually stop making mistakes. And that takes a lot of time. And this is why we are both, I think, careful in selecting students, because I will unlikely accept anyone from, let's say, who's the student, let's say he or she finished third year and he or she has one final year. It's for me, it will not bring much. It may help student for, I don't know, getting a recommendation letter or something like that. But I also need to make sure that I get something out of this. And it takes 2-3 months when students actually go to the lab almost every day. After a couple of months, they actually start coming to their senses and actually become independent. Before that, it's just a constant sort of like, of course, they ask questions. They don't know where things are, and so. But as I said, misconception is that everything is super exciting right away, and that they need to achieve something immediately, right? That doesn't work. It sometimes actually it does. but it's maybe 10% of cases. Yeah.
Do you have any stories with ridiculous, ridiculous events that students did in your lab due to lack of experience or knowledge?
Well, I mean, I guess you want to publish this, and
Nothing
so
ridiculous.
So I probably, if there was something really strange or dangerous happening, I wouldn't say that. But no, I think everything. So, and of course, the lab is. We've been functioning full time 1 year and 2 months. Then COVID came. Okay, and ever since it was kind of like very low activity, unfortunately. But recently, it's not. It's neither funny, it's nor interesting. It's just like, for example, there is a constant shot of distilled water in C4. So we brought actually jugs of distilled water from block 7. There was like 30-liter canisters, huge canisters, which have been used for labs. Okay, and so people started to use that water, and then one of the water was actually a waste. So we didn't know that, but the lab technicians put waste, let's say from labs, I don't know, solutions. Maybe it was like nothing serious, but still there was a lot of salt in this water, but we thought it's a DI water. Some students made gross media, and well, it didn't didn't work really well.
Yeah,
So
I can guess.
yeah.
Okay, and another question in the chat. People ask, how do professors usually pick a topic to research on? So how this process is going?
Well, it's you know, I cannot tell about everyone else, but I can tell about me. Actually, it just comes to you. Usually, I think of the results of the outcomes, and sometimes they are completely unexpected. Okay, and I say, okay, how can I use it to my advantage? In many cases, it's some serendipitous event. For example, in this case, you see blue column, and you ask yourself, what the hell is that? I want to know it. Okay, it's of course very obvious thing to do. In other cases, people have seen these kind of kinetics for a long time, and that's why they were actually suggesting that the DG1 is a deglycase because they thought that they see a deglycation event. They thought that they see this hemithioacetal, and they actually do see that. But in reality, the protein works on free MGO. And because it's an equilibrium, what you see is a hemithioacetal declining, but the protein itself works on free MGO, okay? And so it's a different thing, okay? So interpretation is different. The kinetics is the same. So I thought, okay, because it's not what people think, it's something else. And what is this something else? It's actually a total activity on enzyme. So I'm going to use it for to suggest that this is a new method to measure protein activity. So there are, and here, of course, it's like with synthetic chemistry approaches here, it's a huge serendipitous event that Vsevolod Peshkov works on hemithioacetals. Okay, we talked, and here it would have been extremely nice project if we were able to get enantiomers. Okay, but we get racemic mixtures, unfortunately, in most cases. But because when I told him what I'm doing, he actually told me, it was like almost immediately we said, okay, we need to try this, and it actually works. Enzymes work beautifully on those substrates. It's just we don't get those things separately if they are in a mixture. And then if they are in a mixture, this is almost useless for chemists. We wanted to have pure optical, optically pure compounds. So it just depends. Yeah.
Okay. Thank you.
Yeah.
So I think it was the last question from the chat. It also was the last question for me. So thank you very much for hosting this lab tour. It was very, very interesting. I think many people got many useful notes for them, for their researchers, for their studies. And maybe last question, do you think you can nominate Dr. Andreeva to participate in our lab tours?
I cannot nominate anyone. I will ask her to consider this. Okay.
Of course.
She is not a fan of these kind of activities, but I will insist. So usually it works.
From your words, her lab is also very interesting, and her research is very.
Oh yeah, definitely. It's a high-end research. I mean, it's very sophisticated model system. She asks, questions, the kind of questions she is trying to figure out. people at National Institute of Health. Her competitors are the best people in the world. Okay. So, and of course, she doesn't have a lot of chances, but she still is doing this kind of top-notch science. So yeah, I will talk to her, and I hope that she will make a presentation. Of course, we in all cases like this, we have to ask ourselves what I'm getting out of this, and she that's what she will tell me. But yeah, I'll try to convince her. Okay.
Thank you very much. Thank you very much for the hosting. It was really nice to see you during the COVID times.
Yeah, guys,
professor.
keep up the good spirits, guys. COVID will go away, and everything will go back to normal, hopefully. And you just cheer up a little bit. All right. I know that it's difficult, but I hope to see you guys on campus soon.
Okay. Thank you very much.
All right. All right. Bye, guys.
Goodbye.